Today, I'd like to speak about amitosis in post-embryonic cells, and these are probably due to mycotoxin exposure. I'll be talking about ducks, and I think they meet a challenge that was put forth about 60 years ago. I'll speak about more of that later. I'll also talk about erythroplastid production. Brief review: mitosis, of course, is the part of the cell cycle which is divided into phases known as G1S, G2, and M. The M phase subdivided into interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Ordinarily, the products of mitosis are daughter cells of equal genetic content and size. Ordinarily, that's the case. In amitosis, there is a phase of nuclear constriction, followed by cell membrane constriction, formation of an isthmus. The isthmus later becomes thinned, then cytokinesis occurs, and the daughter cells are not necessarily equal. In amitosis, there are no chromosomes formed, nor is there a spindle apparatus produced. By way of review, some examples of mitosis in duck blood cells. Up here in the upper left, we see a cell transitioning between interphase and prophase, where the chromatin becomes condensed into chromosomes. A little later, those chromosomes assemble at the metaphase plate, getting ready for division. Here is an example of a we call a typical heterophyll in metaphase. It's surprising to some that heterophylls possess the ability to divide. Here's a control cell not dividing for comparison, a reactive lymphocyte, atypical erythrocyte, and so on. A little later in the division cycle, the chromosomes separate, as you see here. This is known as anaphase, of course, arriving at the opposite poles at telophase, and then the constriction of the cytoplasm usually starts about that time. Here we have some cells that are along in the cytokinesis phase. The chromosomes are beginning to reform into nuclei, and there's a slight asymmetry of those daughter cells. These photos come from Lucas's treatise on chicken blood cells, and they indicate that the embryonic red blood cell is a different shape than in the mature animal. The chromatin is usually open. So these cells seen in the embryo are known to be capable of amitosis. Here is one that's well along in that process. The daughter nuclei still remain attached through an isthmus. The cytoplasm is roughly the same size, so this would be a symmetric type amitosic cell division. This cell over here shows the results of an amitotic process. It happened a couple of times. The daughter nuclei are obviously not of equal size, and in this case, one nucleus failed to complete the amitotic division. So Lucas speculated that these things were the property of embryos, and they didn't occur in post-embryonic animals. I will show data to show otherwise. Okay, let's look at some red blood cells in the post-hatch duckling. This one you see here shows clear-cut condensation of the cytoplasm. The nucleus is mostly condensed, but when separation is completed, the one product will contain a nucleus, the other will not, and that will become an erythroplastid. So this is really not an example of amitosis, but a similar phenomenon called erythroplastid production by the cytokinesis process. Okay, now we have some, again, some red cells of circulation. Here's some young ones and some fully hemoglobinized cells, and these others. And this one, we see a clear-cut condensation or constriction of the nucleus, no constriction of the cytoplasm yet. So this would be an early stage of amitosis. Okay, some other examples of amitosis at the early stage in a duckling of 2 weeks of age duckling exposed to AFB1. This polychromatic erythrocyte is clearly in the process of an amitotic division. We see the separation of nascent daughter nuclei. Chromatin is not fully condensed here. It looks like it will separate equally, as will the cytoplasm, approximately. I draw your attention to this fully hemoglobinized erythrocyte shown by the arrow. This is an umbilicus, which is a scar that occurs after a separation of an erythroplastid. So the two processes, erythroplastid production and amitosis, can occur simultaneously in here a 2-week-old duckling exposed to AFB1. From an older duck, this one was waiting to get into the breeding program, and it came from a flock in which we detected a fungus, which I called in 2015 Hemomycetes avium. So here we see a cell in the process of amitosis. Constriction of the cytoplasm and constriction of the nucleus are evident. It looks as if it's slightly asymmetric in this example. One nucleus will be smaller than the other. Here are 3 reactive thrombocytes that are also present, all due probably to the exposure to the fungus. The inset comes from another duck in the same flock. Okay, amitosis midway now. We have separation of the chromatin, and here it's about equal. The daughter nuclei still remain connected through that isthmus, and those cells will be a little on the small size, so they likely could be called microcytes, but that's a midway along in the amitotic process, again in a 2-week-old duckling, well beyond the post-hatch stage. A little later we have an example of further amitosis. We have thinning of the isthmus. We have about equally sized daughter nuclei, and they're more or less condensed. There's never any formation of chromosomes or a spindle apparatus. The daughters here, of course, are going to be microcytes, smaller than the average cell. Note also the presence of atypical red blood cells that occur in blood specimens from ducks that are exposed to AFB1 and have fungal disease. Okay, a look here at some circulating duck erythrocytes. This one is pinching off its cytoplasm. That will be a small erythroplastid. A large pyrenocyte is what that is called. Here's another example of a pyrenocyte that was produced from a fully hemoglobinized cell, and its chromatin was fully condensed. So here is a younger cell over here, polychromatic erythrocyte. Here's an erythroplastid that came from a fully hemoglobinized cell, probably whose nucleus looked like this one. Not amitosis, but erythroplastid production, a similar process. There's such a thing now as complex amitosis. This occurred in a AFB1 exposed duckling that was 2 weeks of age. This happens when a cell divides by amitosis and produces an erythroplastid at the same time. None of these are artifacts. These are all true examples of unusual division of the cells. Notice in this case that the chromatin is not fully condensed. The daughter cells will be unequal in size and in chromatin content. One will be a pyrenocyte, the other will be a microcyte, and the third daughter will be an erythroplastid with an umbilicus. Again, note the presence of an atypical erythrocyte reminiscent of the embryonic erythrocytes described by Lucas way back in 1961. Okay, by way of summary and conclusions, Erythroplastids and amitosis can both occur in mature and immature red blood cells. There may be a common pathway partially, but each process has some unique features. What causes it? Well, I'm speculating here that it is due to a degree of damage to the DNA, probably caused by exposure to mycotoxins. The process can be complex, producing both an amitotic division and an erythroplastid production at the same time. A good question is whether these processes can occur in leukocytes. This is under study, but I do have some supportive evidence to show that at another forum. And I think in general this fulfills Lucas's criteria for demonstration of amitosis in post-embryonic avians. Thank you very much.